Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hello, everyone. This is Jason Key from SB Grid in Boston. Thanks for joining us today. This is the SB Grid webinar series. Two more webinars in this series coming up. We've got in April, we've got Dmitry Taganov from Genentech, who's going to tell us about WARP, which will be very exciting. That was a tool that was primarily available only for Windows for a long time, and it was made available for Linux and has become much more widely used. So I know that we're using it locally at HMS. Lots of other groups are using it. It would be great to hear more about it. It's under rapid development, new versions all the time. And then in May, we've got Ellen Zong from Princeton, formerly of MIT, who's going to be talking about Dragon AI. So Ellen, I think, kicked off CryoDragon when she was at MIT, which I think also led to the development of TomoDragon, and since moving to Princeton has released Dragon AI. So it'll be exciting to hear about Dragon AI. That's the last two in this series. If you have thoughts, suggestions, titles you want to hear about, we're always interested in hearing for requests. We'll start lining those up for the next series in the fall. So reach out, let us know. We will try to line up those titles by request. Otherwise, we have plenty of great titles to pick from. So we'll be lining up our new series soon. With that, it is my pleasure to introduce Genki Terashi. So Genki is joining us from Purdue University in beautiful West Lafayette, Indiana, my alma mater, where I was an undergraduate. So I was lucky enough to be able to visit Genki in his lab in the Kihara lab this summer. I was back in Lafayette, and we got to talk about all of the software projects that are going in their lab. Many of these are focused on structural biology applications, primarily cryo-EM, but surely not all. There are many software projects going in the lab there. So today, Genki's going to tell us about two of those, Deep Main Mast and DAQ. So Genki, go ahead.
Thank you for the introduction. So first of all, I would like to thank the SB Grid team for inviting me to this webinar. So this is a very really wonderful opportunities to present our modeling tools and model validation tools for the real users. So today I'm going to talk about our modeling tool, Deep Main Mast, and DAQ core. Okay, so these are today's content. So first, I'd like to introduce how the Deep Main Mast works. So Deep Main Mast workflow, and also I want to present the performance of Deep Main Mast, and I will show how to use the Deep Main Mast programs on the SB Grid package. I will demonstrate the command and how to visualize the result by PyMod. And also I will show the DAQ score. This is the AI-based model validation method. Then I would like to show the performance of the DAQs, then also how to use the DAQ score in the SB Grid package. Okay. Okay, so this is a Deep Main Mast workflow. Let me explain a brief overview of our de novo protein structure modeling. So suppose you have a density map and sequence information. Then Deep Main Mast can build the atomic model from the density map to the atomic models. So first, these EM maps are converted to the local structural features using the deep learning method. So we use a 3D unit architecture. It is a very good architecture to segment out the individual regions for the features. For example, using this 3D unit, the DeepMind must automatically identify that what kind of the atom type are existing in each voxel. So this is an example of the output files. So from the input density map, DeepMind must identify that the alpha carbon positions like green beads. And also it identified that the nitrogen position, backbone nitrogen positions, and oxygen positions. It also can predict the amino acid types. So this is a example of the output. From the density map, it can predict what kind of amino acid type, 20 amino acid type are existing. Each color representing the different amino acid type. So for example, blue is glycine, then this orange is a tryptophan, like so on. Based on these amino acid type predictions and atom type predictions, then DeepMind must connect these information as a fragment of the protein structure. To connect these alpha carbon positions, so DeepMind must perform the vehicle routing problem solver. So vehicle routing problem solver is a kind of the traveling salesman solver. So connect the green dot, then find the optimal path that covers the entire carbon position, alpha carbon position. At the same time, so DeepMind must align the amino acid sequence based on the predicted amino acid type predictions. Then it can generate a lot of local fragment structures. So here we can see that the helical structure is predicted, but some connection is not accurate. Then based on the generated fragment, it combines into the single PDB files, multi-chain protein structure models. Then it can finally get the final models. So now we can see that's the very clear alpha helix structures and nice loop connections. Then everything looks well packed, and also the sequence amino acid assignment are performed. So DeepMind must have another interesting features. It can consider the additional information such as AlphaFold 2 models. So nowadays, many researchers are using the AlphaFold models, so because the performance is good, and also the local structure is very accurate. So DeepMind can consider these additional informations, AlphaFold 2 models, as a fragment, then improve the performance. So even the AlphaFold 2 models, sometimes domain orientation is not accurate, or global structure is very different. But so DeepMind must consider the local features and the local alpha carbon positions, then finding that's the corresponding positions. So for example, AlphaFold 2 model looks like this, then it automatically identify that DeepMind must automatically identify that this domain is corresponding to around here, here. Then this alpha helix cylinder shape might be corresponding here. Then combine the all fragment into the final models. So this is a DeepMind architecture. It's very simple. So predicting the local feature by using the 3D unit, then connect these points by using the vehicle routing problem solver, then assemble the fragment into the one final random model. So we perform the benchmarking using the single chain modeling. So I want to show some interesting examples. So this is one of the examples in our benchmarking data set. So resolution is quite high, 2.6. So for such resolutions, so DeepMeMath can trace the backbone positions very accurately. So DeepMeMath space means just doing the backbone tracing without using the AlphaFold2 models. So this pink ribbon structure is a deposit PDB structure. Then this rainbow colored model is a predicted models. So as we can see that these pink structures and rainbow structure models are fitted very well because resolution very high and it can trace very accurately. So this is a result from the DeepMeMath full pipeline. So full pipeline means that it considers the AlphaFold2 models as additional information. Yes, so DeepMeMath achieves a very nice result. Interestingly, for this target, the AlphaFold2 cannot predict the accurate models. So because it consists of two domains, and this part, so beta rich domain looks fine, well fitted, but these domains are kind of twisted. That's why the RMSD value is a 10 point something. But even such case, so DeepMeMath automatically identified that each AlphaFold regions are corresponding to the density, then assembles the entire structure. These are other examples. It has a very long beta sheet structure. So DeepMeMath based tracing method and DeepMeMath full pipeline AlphaFold using the AlphaFold models can achieve the very nice result. Even the AlphaFold2 generates the strange loop here, cannot build these regions, but it can build a nice structure. Because DeepMeMath use big routing problem solver, it can handle the multiple chain modelings, oligomer modelings. So this is one of the examples in our benchmarking data set. It has 5 chains. Then this is the input density map. And these colors are based on the deposit structures. So this is the deposit structures and the input density map. So DeepMeMath can build the entire structures and also can assign the chain IDs very accurately. So corresponding chains are matched very nicely, 5 chains. For comparisons, we also performed DeepTracer. So DeepTracer is another very popular modeling tool. It's very fast and also relatively accurate. So yeah, DeepTracer builds almost all regions nicely, but sometimes it fails to assign the chain IDs. For example, this green chain A positions, it's split it into the different chains. And this part is some different chains are existing. In the similar result happens. So in this target, so this is the input density map, and this is the DeepMeMath models, and this is a DeepTracer models. So DeepMeMath can assign the chain IDs very accurately. Okay, so we check the performance among the 178 targets, then compare the DeepMeMath space, only the main chain tracing method, and DeepMeMath full pipeline using the AlphaFold2 models, and CII Taser. This is another state-of-art modeling tool that use the template-based modeling, and AlphaFold2 itself. So I use the TM score as an evaluation matrix. So if TM score is a 1.0, so that means this modeling is perfect, 100% accurate. The higher is better. Then I first compared the CII tether as the x-axis, the y-axis is the deep main mass. The AlphaFold2 x-axis and y-axis the deep main mass space. So basically, it's almost similar result with the CLI tethers and deep main mass base. It's a bit better. Then deep main mass base is almost same performance with AlphaFold2. So because these bottom regions, lower TM score of the deep main mass base is a resolution is relatively low. So local resolution is low. In that case, the tracing doesn't work well. Cannot find the connections between the CLF atoms. So that's why there are some cluster around here, low TM score. But CLI tether is using the template, so available PDB structures. So in that case, this CLI tether can achieve the better result than deep main mass base. And also, AlphaFold2 doesn't use the cryo-EM data. So AlphaFold2 also achieved a better result. But if the deep main mass consider the AlphaFold2 models, then performance are significantly improved. Because the deep main mass considered AlphaFold2 models, then it can fill the locally low resolution regions. So significantly, the deep main mass full pipeline achieves a better result than CLI tether. Then deep main mass full pipeline achieves a much better result than AlphaFold2. So one of the common question is that what is the resolution range for the deep main mass? So short answer is that if the map resolution is better than 5 angstrom, deep main mass can build good models. So in our papers, we carefully check the local resolutions for each CLF atom positions, then how the local resolutions affect the modeling accuracy. So this is a CLF position accuracies. So this is a recall. So how much the positions of the native structures are covered by the deep main mass models. Then blue is a deep main mass space, just backbone tracing method. Then deep main mass full pipelines, considering the AlphaFold2 models. Then if the resolution is better than the 3 angstrom, local resolution is better than 3 angstrom, then 99%, almost 99% of the positions are predicted. Then 3 to 4 angstrom resolution, local resolutions, 85% are predicted. Then 4 to 5 angstrom local resolutions, around 70% positions are predicted. But if the resolution is worse than 5 angstrom, then performance dramatically drops. So only half of the positions are predicted. Then when we compare with the deep main mass base and deep main mass full pipeline, of course, using the AlphaFold2 models, AlphaFold2 models help to improve the coverage a little bit, 10%, in this case, 20% improvement. So using the AlphaFold2 models can help deep main mass to achieve the better prediction. So this is another metrics, amino acid type assignment. So how many positions are correctly assigned amino acid type? So if the resolution is better than 3 angstrom, local resolution is better than 3 angstrom, then 99% of the positions are correctly predicted. But if the resolution is lower than 5 angstrom. So deep mass base couldn't assign the amino acid type correctly. So 60% or 50% among the modeled regions. But if we use the AlphaFold2 models, it can maintain the high precision values, amino acid assignment accuracies. So this is a typical examples of how the AlphaFold2 models can help the deep mass performance. So this is a 4.1 angstrom resolution, but the local resolutions are very different. Even the map resolution is 4.1, but some regions are completely disappeared. The pink structure is the PDB deposit structure, and the rainbow model is a predicted models. So deep mass base is try to trace the entire regions, but the N-terminal regions here, then go to here, then there are some strange connections. Then TM score is 0.66 and RMS is 12. It's not good models. So this part, so local resolution is quite low. So that's why the deep mass cannot build the helical structure. For these targets, so AlphaFold2 works very well. So TM score is 0.92. Overall structure looks similar, but interestingly, some shifting happens. For example, this helix, red helix regions, it's shifted up, but this yellow helix aligned very well. Then also these blue helix regions are slightly shifted. So overall structure looks nice, but for this target, AlphaFold2 models are slightly wrong relative orientations on the alpha helix positions. Then combining these two tracing and AlphaFold2 structures, deep mass full pipeline fix the problems. So it can build a very nice structure, fitted very well to the density map, and also even the local resolution is low, but it can fill the loop regions because it considers the AlphaFold2 model. So this is the advantage of using the AlphaFold2 models. Even the local resolution is low, it can assign the amino acid type correctly and also fill the missing loops. Okay, so then the deep mass is available in the SB Grid. So our code is also available on the GitHub page. So if you want to use the deep mass on the SB Grid, please try to type the SB Grid command line, the info deep mass. Then if these informations are shown, then your system already has a deep mass. Then I'd like to show how to use the deep mass on the SB Grid. So to use the SB Grid command line, so please type the SB Grid CLI command line and shell, then activate the SB Grid shell environment. Then specify the GPU IDs. So for example, the CUDA visible device equals 0. So that means that I want to use this GPU number 0. Then there are several options. So for example, if we want to trace the backbone position, so if we want to get the result relatively quickly, please try to use this command. So -f, then you can specify the faster format sequence file, then -m, the EM file, then P, the program path, then then -C, then control levels, the -O, the output directories, the -T, and tracing time. Large T is the assemble time. So if the target sequence and density map is relatively small, you can use a small number, like let's say the 60. But it has a lot of chains and volume is big, then please use the 600, sometimes 1,000. It depends on the size. And also DMMS can build a Flaton model using the Rosetta package. So to do that, please add the -X, then put the Rosetta program path. Then it automatically builds the side chains and define the side chain orientations and also fill the missing loops using the Rosetta program. Later I will show the exact command. If we want to use AlphaFold models as additional information, please specify the -A option, then put the AlphaFold models in the PDB form. Then DeepMS automatically consider the AlphaFold models as additional information. Okay, so this is the actual command. So last night I tried the same command. So backbone tracing using the DeepMS. So command is this, then specify the FAST format file. So FAST format file looks like this. So each chain, so this is the entry ID line, then sequence information, the ID line, the sequence information, ID line, sequence information. So this case, the 3-chain heterodyne. Then specify the density map. Then also you need to specify the -P and program path. Then also the user can specify the control level 0.1. If you are not sure what is the control levels, you can put the 0.0. So because the deep learning architecture automatically detects the C alpha positions, nitrogens, carbon positions. But if you know the good control levels, then please specify. Because using this control level, it can significantly reduce the computational time. Otherwise, unit architecture deep learning part can need to scan the entire voxel region. Okay, then Flaton build modelings using the Rosetta. So please use this -X options. So in the SBI grid, so Rosetta is also available. So please try to type the SB grid CLI, the info Rosetta. If you show up if this command show the place of the Rosettas or it's already installed, then you can use maybe this path. This specify the path of the Rosetta program. But if you don't have a Rosettas, or if you already installed a Rosetta package locally, then you can specify that directory. For example, in my case, I also installed this version of the Rosetta. So then home bin, Rosetta bin, Linux, like so on. Then it can build the model. Okay, so this is a brief introduction of how to use the command line. Then let me show the terminals. So maybe you can see the terminal here. Then first, SB grid shell. Then it will activate the shell command, shell script, shell environment. Okay. Okay. Then I already set up data. So for example, this is a FASTA file and MRC file. Then command looks like this. So this is a deep mass shell script. The -f, then specify the FASTA file, the -m, they specify the map file, the -b, they specify the path of the programs, then control level, then output directory. Okay, okay, first I need to specify the GPU ID. Yes, it's running. Okay, then meanwhile,
Genki, one quick question while you were specifying the GPU ID. Is it possible to use multiple GPUs or should you specify a single GPU?
it's a single GPU. So yeah, because my environment, I have a multiple GPU cards. So then GP1 is a very old. It's just for display the monitor, so it cannot compute the deep main math. So that's why I need to specify that the specific GPU ID.
All right, great. Thanks.
Okay. Okay, so then, yeah, so the -a option, if we want to use ALPHA2 models as a reference, as a additional information, then please use -a option. Okay, then let me explain that what kind of output files are generated. So in the output directories, so there should be the deep main math.pdb. This is the final predicted structure models, and also the result directories. So for example, in the result directories, there's a unit directories. So this unit directory contains all predictions computed by the unit. So this is showing the list of the unit directories. It has an atom, GB, background, and alpha carbon, C alpha, carbon, and C beta, carbon, nitrogen, oxygen, and others. So these information predicted results are stored in the MRC format file, atom type prediction. And also each amino acid type predictions are also recorded in the MRC file. So if you are interested in the position of the alpha carbons, maybe you can visualize using these MRC files. So this is the actual examples. So gray is the input map. Then green is the alpha carbon positions. Then everything normalized by the probability values, so 0 to 1. Then I use a 0.5 cutoff values. So that means that these green regions are predicted as over 50% of the chance there is a alpha carbon. Then blue is the nitrogens, backbone nitrogens, and orange is oxygen atoms. Okay. Then also deepmainmath.pdb. So this is the final model from the deep main math protocols. It contains 2 or 4 models. So if you perform only the backbone tracing command, it should contain only the C alpha models, model 1s, and C alpha models, model 2s. So model 1 is entire regions, entire predicted regions. Then model 2 is only the reliable regions. So because sometimes the deep main math predicts the structures as not confident, is low confidence. So in that case, the model 2, these regions are removed. If you perform the full atom building pipelines, so using the -X Rosetta, then it should have a model 1, 2, 4. Model 3 is a full atom model. The model 4, again, the same full atom models, but it removed, low confident regions are removed. All structures are already scored by the DAQ score. So next part I'll show that what is the DAQ score, but it's already scored by the DAQ score. Then using this command, you can visualize the model and also the colored by the DAQ score. Okay, let me show the examples. Okay, this is the output files of the DeepMainMath. So this DeepMainMath.pdb contains 4 models. So but this panel is corresponding with all of the 4 models. It's not useful. Then I can type the split command. Split state DeepMainMath. Then now you can see the models individually. This is the model 1, model 2, model 3, model 4. Yeah. Model 1 is a C alpha model. Then model 3 is all of the full atom models. Then colored by chain. Then the 3 heterogonals, 3 chain. Then here you can type the spectrum B, red, white, blue, then O -1 to 1, it's colored by the confidence score. So blue region is at the DAQ score, so confidence score is very high. But the red regions is a bit low because of low resolution and so on. So from the DeepMainMath models, so you can find which region is reliable. Almost all regions are reliable, but some regions are not confident. C alpha model and full atom model. Okay, yeah, now your program is working. Okay, so yeah, this is the DeepMainMath. So it's very easy in the SB Grid command line. It's only one command. You need to specify that the FASTA format files and density map. If AlphaFold2 models are available, you can use AlphaFold2 models. It's very easy.
Quick question here.
Yes.
Your DeepMath, the output PDB contains 2 to 4 models from every run. So, and those are condensed within the same file. So it might not be apparent to users who sometimes consider like a PDB to be a singular model that there are multiple models within the same file. So using a tool like PyMol, you can see them just by like splitting them. So it's yeah, I just wanted to note that that file contains multiple models. So that's where PyMol really comes in handy, and you can like visualize the multiple. It does on that ensemble view very nicely. So yeah.
Okay. Yes.
You say 2 to 4 here. Does that mean in some cases the full model's not in that output file?
Oh yes. So if the user specified only the backbone tracings, it contains only C alpha models, model 1, model 2. But if user specified as the full atom construction -X option, then it should contain the 1 to 4, 1, 2, 3, 4.
That's with Rosetta, when you add the Rosetta part.
Yes.
Yeah.
Yes.
All right. That makes sense. Great. Thanks.
Okay. Okay, then let's move on to the DAQ scores, so deep learning amino acid-wise model quality scores. So suppose, you have a density map and atomic models. So DAQ scores can predict the local properties using the deep learning method. Let's say that this position is corresponding to the alpha helix and beta secondary structures, or nitrogen, oxygens, and also the amino acid type. So atom type, 20 amino acid type, helix shape, coil, secondary structure type. Then by considering this predicted local feature and input PDB structure, it can identify that how much that these provided atomic models agree with the predicted local features. For example, if these positions are predicted as a tryptophan by the deep learning method, but the models have a glycine, then it disagrees. It's not agree with our predictions. Then DAQ score said it's not consistent. It's not agree. It doesn't agree with the local density feature. Then flag that these positions might be the wrong structures. This is the DAQ score. So DAQ score is trained using the available density map and PDB structures. So it extracts the 11 by 11 voxel from the density map, then using the level based on the atomic models. So atomic models have amino acid type and atom type information and the secondary structure information. Based on these informations and local density 11 by 11 by 11 voxels, it can try to predict these level information. So this is a DAQ score. From the local density environment, it predicts the amino acid type, atom type, secondary structure type, then check the agreement, consistency between the input PDB file and predicted local feature. Why this DAQ score is so important? So because the DAQ can identify the potential misaligned edges in PDBs. So during this project, we noticed that even the deposit PDB structure, it had some inconsistencies. So for example, this is the actual PDB, 6L54 and 6Z3R. These two entries have an exact same sequence. And resolution is a bit different, but overall structure looks very similar. But interestingly, this region, so this cyan structure, region number 425 and 423 modeled here. In the pink structures, 6Z3R, region number 425 modeled here, and 423 modeled here. Even the same protein, same sequence, but the two deposit PDB entries have a different modeling. So that means one of them have a misaligned, wrong models. We don't know. So in such case, if we use a DAQ score clearly say that these red regions, the low DAQ score, this position is wrong. It's not agree with the local density features. So by using the DAQ score, we can identify that's the misaligned region, even in the deposit PDB structure. That's why the DAQ score is so important to validate the atomic model. So we benchmark our DAQ score using the PDB entries, which has multiple versions. So multiple versions means that the author deposited the PDB atomic model the first time, then later the author revised or updated the coordinate. So this is the first version, first deposit models, and this is the revised models. So that means that by comparing the first version, first deposit models and revised models, we can find that which positions are updated by the author. So these inconsistent positions means that these magenta regions are updated by the author. So that means that in the first version, the author make a mistake to assign the sequence. Then we apply the DAQ score to the first version and revised version. Then clearly we can see that in the first versions, these red regions have a low DAQ score. Then actually this region is clearly corresponding to the inconsistent positions. Inconsistent positions means updated regions. The author updated the coordinate and sequence assignment as these magenta regions. That region is clearly corresponding to the first version at the low DAQ score regions. So this is another example. So these magenta regions are updated by author. Then in the first version of the model, DAQ score says these positions are not accurate. Yeah. We check all of the positions, updated positions, and not updated positions. So that means the not updated position is correct, good, then not have to be updated. But the inconsistent regions, red positions, is updated regions. Then check the DAQ score. And clearly, we can see that in the updated positions, DAQ score, amino acid assignment score, is lower than 0. If the score is negative, especially the DAQ score is lower than -1, then it's highly possible that this position is not accurate. But if the DAQ score is higher than 1.0, it's highly possible that this position is accurate, correct. So currently, the model validation tools, so there is a Q-score. This is one of the most major model validation tools. So we compare with the DAQ AA score, amino acid base score, and Q scores. Then found that the DAQ score can identify that incorrect regions, misaligned regions, more accurately than Q score. So we use average AUC, area under curve, ROC curve, and also area under curve of the precision recalls. And because the Q score have a different window settings, so one residue window, 3 residues, 11 residues, 19 residues windows, we use the same window size, then check the performance of the identified at the misaligned regions. Then for all of the window size, we achieve the clearly better performance than the Q score. So because the Q score is measured how well the atomic model fit to the local densities, so it cannot consider the local environment. But DAQ score consider the local environment, and also it can considering the residue interactions. That kind of information are also captured by the deep learning. So that's why the DAQ can identify the misaligned regions very accurately. So currently, yes, the experimental groups start using our DAQ score to validate their models. So this is a Nature Communications papers. So they check the M4 muscarine acetylcholine receptor structure. Then these two structure is the same sequence, same molecules, but different map and different conditions. Then this red structure has a relatively low DAQ score because the resolution is not accurate and modeling is some mistake happens. But they achieve the very nice modeling. Then they can achieve the very nice DAQ score, positive DAQ score. So DAQ scores can be used as a validation tool. This is another paper. So they build atomic models, large atomic models. Then they demonstrate that the overall region looks very accurate. Then it's consistent with local cross correlation information, local cross correlations. So this is another paper, Nature papers. So this author used the DAQ score, then confirmed that their predicted their models, their protein structure models have a positive DAQ score, confirmed that their model is reliable. And this Nature Structural Molecular Biology papers, then they computed the DAQ score for individual domains, then which domain regions have a relatively low DAQ scores or high DAQ scores. So they highlighted that these regions and some positions have a relatively low DAQ score. So that means that these regions have not enough information for the modeling, and map quality also not good. Okay, so then again, so DAQ program is available in SB Grid. Please confirm that the availability in your system. Please type SB Grid CLI info DAQs. Then this kind of the informations are showed up. So currently the latest version in the 1.1.0. Please check the 1.1.0. This is the latest version. So this program is very easy. So first, activate the SB Grid CLI shell. Then you need to type the DAQ copy commands. So this DAQ copy command copy the entire package to the local folder. Then move to the DAQ copied directories. Then type this command. Then it automatically compute the DAQ score. In the DAQ directories, so there is a predict result directory. Then it should have a subdirectories based on the EM map. Then it has a DAQ score window 9 w9.pdb. This file contains a result of the DAQ score. Okay, this is some tips. If you want to use a very large density map, so it is good to resize the grid space, the 1.0, using Camera X. So our script also contains the resizing script, but it's relatively slower than Camera X. So if you are using the Camera X and also want to compute the DAQ score using the very big density map, please try to use the Camera X. So this command is also available in the GitHub page. Please later, please check the result, GitHub page. Then again, so for visualizing the DAQ score using the PyMOS, please type the spectrum B, red, white, blue, DAQ score W9 is a model name of this panel. Then -1 to 1. Then red region is a low DAQ score. Blue region is a high DAQ score. Okay, let me quickly quickly show the command. Okay, modeling is done. So here, there's a DAQ directory, then change DAQ directory. I already copied the file, this file, then python.daq, then main.py, the -f, they specify the MRC files, then specify the GPU ID here, then -p, the protein structure. That's it. Yes, it automatically generates the predicted result and this folder. Okay, let me quickly show how to visualize the result of the DAQ score. So this is a DAQ score window9.pdb file. So this is a spike protein, the COVID-19. Then let's type the spectrum B, red, white, blue, O. Yeah. Then it shows the DAQ scores. Blue region is a reliable regions, high DAQ score regions. Then white is a 0. Then red is a negative DAQ score regions. So clearly we can see that these beta sheet regions have a negative DAQ score. It's highly possible that these positions have a misaligned regions. Okay, let me quickly align split chain. By using the split chain command, you can visualize the individual chains. Then I can superimpose all chains into the chain A. So interestingly, this target 7FET is homotrimer, same sequence, almost same structure, but only the chain B have a clearly low DAQ score. This region. If we focus on the, for example, cysteine here, 605, show 6. These two residues are clearly shifted. Cysteine is clearly shifted. Yeah. 605, 605. Only chain B have these positions, and chain A and C model the cysteine, serines 605 are modeled here. It's highly possible that somehow the author build the 605 positions here, but it should be here. Okay, like this, so even in the PDB entries, so we can verify the accuracy of the PDB structure using the deep learning method. Okay, okay, this is additional information. So our program is also available in our server. So if you don't have a GPU, please visit our web server. So emkikalab.org. It's free. Then you can register the IDs and accounts. Then you can store the all results. Please try to use DeepMember server. And also DAQ score server is available. You just put the PDB structures and CryoEMF upload. Then you can get the same result. And if you want to compute some specs, compute the DAQ score for some specific PDB entries, we are also releasing the DAQ score database. So currently we have 231,000 entries. So it's pre-computed DAQ score. So if you want to check the accuracy of some specific PDB ID, please try to use DAQ score database. Okay. Then, yeah, so I'd like to thank the Professor Kiharas, our PIs of our lab, and also I want to acknowledge the Xiaowan who developed the deep learning bars, and also the Debashis and Tsukasas. So they also help us to build these wonderful tools. So additionally, I'd like to thank our collaborators and the funding and the support for the support of this work. So finally, a special thanks to the SB Grid consortium for providing this platform. Okay, so thank you for your attention. So please leave your comment or questions in the chat box. I'm happy to answer any questions.
Thank you very much, Kiki. That was fantastic. Questions, you can raise your hand. If you click the raise hand button, you'll pop up to the top of the list and I can unmute you and you could ask your question. We have a couple here. One coming in for, I'll kick it off real quick. Is DAQ computationally tractable to refine models against? Could you do like a molecular refinement in a sort of conventional refinement sense like you would in, say, I don't know, Phoenix or something like that, but use DAQ as your per residue refinement metric?
Yes, actually, we also developed the DAQ refine. So because the DAQ can identify that which position is wrong, which position is correct, then we use that information to the AlphaFold. So we combine the DAQ score, the AlphaFold, then define the structure. So we already released that code. So today I didn't show that.
Okay, great. Yeah,
we have.
That seems like it would be very, very useful for making these, sort of corrections on, before they get deposited. Ahmed, let's go ahead. Let me, you should be able to unmute.
Yep.
Go
Great
ahead.
talk. Thank you, Genki. My question is, can the software handle symmetry, protein symmetry?
Okay, yes. So are you asking about the DeepMinMask?
Yes.
DeepMinMask. Okay, so currently, it cannot handle. It doesn't have a functions on the symmetry. So yes, short answer is this. So you need to specify that the number of the chains and map information.
I had a similar question regarding inputs. What about inputs of ensembles? Like if you had AlphaFold 2 models, AlphaFold, you could have an ensemble of models. Is it useful or possible to take an input of model ensembles?
So currently, DeepMinMask consider the one structures for one chain. But later, we are planning to use the ensemble of the structures, so different candidate of the models. So it's very good idea.
Other questions? Let's see. Great. Well, thank you very much, Genki. Since we're about at time, we can leave it at that. But if you have any other questions, reach out. DeepMinMask and DAQ are in SB Grid. There is also a server. There is a collab. There are, and great resources. And I think the lab as a software development team is super responsive and wants people to be using the software and getting good results. So I'm sure they would be happy if you reached out. And with that, thank you very much. And thank you everyone for joining today. Be sure to catch us next month.